The Business of Biotech is produced by Life Science Connect and its community of learning, solving, and sourcing resources for biopharma decision makers. If you're working on biologics process development and manufacturing challenges, you need to swing by bioprocessonline.com. If you're trying to stay ahead of the cell or gene therapy curve, visit cellandgene.com. When it's time to map out your clinical course, let clinicalleader.com help. And if optimizing outsourcing decisions is what you're after, check out outsourcepharma.com. We're Life Science Connect, and we're here to help. The concept of programmable biology is fueling a new breed of biotech, one that requires the marriage of computational and traditional scientists at every step of the journey, from discovery to commercial. Bit.bio, spelled Bit.bio, is exemplary of this new breed. The preclinical cell therapy company is developing therapeutics in metabolism and endocrinology, immunology, and neurology, and its founder and CEO, Dr. Mark Kotter, doesn't pull any punches when addressing the complexity involved in building out the company's capabilities. On the discovery side alone, Bit.bio has hired and integrated stem cell biologists, synthetic biologists, genetic engineering experts, cellular biologists, sequencing experts, data scientists, bioinformatics pros, and machine learning experts. I'm Matt Pillar. This is the Business of Biotech, and I caught up with Dr. Kotter in San Francisco to talk about the work Bit.bio is doing, how it's doing it, and how he and his leadership team are recruiting and retaining a new breed of biotech talent to sustain the effort. Let's give it a listen. Dr. Mark Kotter, founder and CEO at Bit.bio. So let's start right there, Bit.bio. What's behind the name?
It's really the convergence of data science and biology. We are dealing with a paradigm of synthetic or programmable biology, which has opened up opportunities and solutions to long-standing problems in turning pluripotent stem cells into different cell types. So we're really a cell manufacturing, creation, and therapy company. Mm-hmm. And this new paradigm, this programmable biology, provides a solution to consistency, scalability, but also even functionality of the cells.
Yeah, okay. And you remain currently a clinical scientist at the university?
Yeah, my background is actually, I'm a neurosurgeon and a stem cell biologist. And I had naively the idea of developing cell therapies. I didn't know how hard this is actually when I started out. And it's only because we were able to assemble a fantastic team that we're actually now doing it. Mm-hmm. The ambition always was, to extend, my medical practice to something broader.
Yeah, that's interesting. You didn't know how hard this was. You were a neurosurgeon.
That's right.
Yeah. How much harder than that could it be?
Well, I think the complexities are very different, let's say.
Yeah.
Yeah.
What inspired you to become an entrepreneur?
It was the realization that the means that I have in academia would not allow me to really tackle this huge problem. So there's two things really. Cell therapies for spinal cord injuries were my initial inspiration. I see patients that whose lives change at a sort of flick of a switch. Mm-hmm. And the only thing neurosurgeons can do is put the bones together, but there's not much we can do to enhance recovery. And so that was really the driver. And I thought small molecules, biologics won't do it. It needs something much more complex. And there's been really interesting studies in this field with cell therapies on the preclinical side, and we've seen some clinical transition. Now, the problem with cell therapies is that they're extraordinarily expensive. Quite frankly, even now, those that work in oncology, it's not a good business model. Some of them are still loss-making drugs. Mm-hmm. So that needed to change. That brought me into the idea of pluripotent stem cells. And then also I realized how incredibly difficult it is actually to control these cells. And the second big thing that hit me was when I was doing my research, like everyone else, we were using mouse rat cells and models. But as a neurosurgical trainee, I was able to access human biopsies. So comparing specific cell types of interest, I found out that they look the same, but biologically, there's a huge difference. So, growth factors that do one thing in the rat and the mouse have a complete opposite effect on the human cell. And for me, that was like another sort of huge sort of inflection point, because I thought, wow, if I continue what I've done before, I'm going to be a fantastic mouse doctor. That's really what I want to be. And so, and this really sort of, brought me to enter this space. Yeah, and little did I know that we would be able to, find a piece of biology that is really incredible, actually.
Was Bit.bio your first foray into entrepreneurship in biotech?
It sort of happened a little bit at the same time. We spun out now 3 companies out of the lab. Bit.bio is the one I'm sticking to. I'm running. There's another company called Meatable in the cultured meat field. So they also need cells, obviously non-human. They need muscle and fat cells. And it turns out this technology is so scalable that they can reach price parity. So that was a parallel thing that happened. The companies initially really worked very closely together. Meatable very focused on scale up, Bit.bio very focused on generation of the fundamental tech. And then more recently, the flip side of iPSCs, of pluripotent stem cells, the in healthy and young state led to a small other spin out called Bio.
Okay, yeah, I've got some questions for you on those spin outs, particularly Meatable, very very interesting company. But I want to linger for a minute on your transition from practicing medicine and being a research scientist to the business, to the biotech business. It's not an easy transition to make.
A lot of things to learn.
A lot of things to learn. So share with me a little bit about that. Like, what was, I don't know, like what were some of the, give me the top, I guess, 3 challenges that you faced in, deciding that you had science that was worth building a business around, but how do you do that?
Yeah. So let me reflect on what's foundationally different between academia and business. In academia is really centered around the individual. And the questions that you ask based on the funding system that is in place are projects. Now, in biotech, I found it's the opposite. It's a team sport. The questions that you ask is, how do I make this work? So these are real big problems that you solve by bringing together, in our case, really very diverse teams and people that would never encounter each other in an academic setting. The resources that it requires, of course, much larger, and the pressure is higher. So I would say there are the big difference is team sport between in biotech and really a very individual sport in academia.
Let me ask you to pause there real quick. When you talk about bringing people together, creating a team, and bringing people together from different disciplines that may have never encountered one another in an academic setting, give me an example of that. Are you talking like technologists or computational people and biologists?
So the thing that we needed to solve to create Bit.bio, which is a platform that can reproduce any human cell type from pluripotent stem cells using transcription factor programming. So let's talk about what that actually means. So you can instruct cell identities by activating the right set of transcription factors. That's the foundational insight that can be traced back to 1980s, Howard Weintraub, and then Yamanaka, of course, for transcription factors, pluripotent stem cell. And then I would really want to shout out Marius Wernick and Tom Sudhoff here at Stanford that have shown that this is a paradigm that seems to be generalizable. So that's the mind, the framework. Now, within this framework has two consequences. On the discovery side, when you think about stem cell biologists, they've been trying to find ways of coaxing cells, let's say along Waddington's landscape, using molecules to stepwise coach them into, a skin cell, a brain cell. The issue with that approach is you never know when you're there. And that's why there's nobody in the industry that has ever created a differentiation protocol from scratch, as far as I know. So industry has taken academic IP and has process developed it, and that's the basis of the many IPC companies that you see. Now, a foundational consequence of this transition to that new paradigm is that you're now when the endpoint is, because you have to find the right combination of transcription factors, and that's going to give you the cell type. So you've got a set of 2,000, 2,500 transcription factors, and combinations of these will create cell types. And that means you can brute force it. You can build a platform that screens screens transcription factors, and a data science platform that reads this out. And you can then sort of approach, and when you build this, you can create new cell types from scratch. And we've demonstrated this in Bit.bio. The other thing that this approach can do is it shortens the time period of the transition from pluripotent stem cell to the cell type by a factor of 10. So, and it's single-step protocols. And the little thing that we developed in Cambridge is using genomic safe harbors, we got to a deterministic paradigm. So we can produce cells, batches of cells that are always the same. So that's the sort of framework. Now, when you think about what do we need to really build out this platform? On the discovery side, we need stem cell biologists. We need synthetic biologists. We need people that are experts in genetic engineering. We need experts that know about the cell types that you want to produce. We need sequencing experts and technologists. We need data scientists, bioinformatics, and machine learning people. So that's sort of the basis of the discovery platform. Now, one thing that we've learned is you put two biologists together in the room, and they're using the same words, and the connotations are very different. Differentiation, stem cell speak, is very different from differentiation, for example, in the immunology field. So we're divided by the same language, and that, so it has from the beginning onwards, we had to really come together and find language to describe what we do and as a shared basis. So that's the discovery part. Then you go into the manufacturing side. Suddenly you've got process engineers. You have, people that know something about assays, bioreactors, and all of that. Again, that's a completely new cohort of individuals with completely different mindsets and backgrounds. Again, those needed to come together. And as a business, then you have to have obviously the commercial side as well. So it's been incredibly complex to bring all these people together and align them on a shared vision, but at the same time give them a language, processes that they can follow in order to build an efficient machine. And I thought it was really fascinating. So we sort of did this very consciously, also in terms of how we shape the culture. I was very sort of what's the English word? I really tried very hard to get to a certain point, and it worked.
How did you prepare for that? Like you talked about when you were in academics,
Yeah.
you're sort of an island unto yourself.
Yeah.
And now you've got diverse teams of many people doing many different things, but having to do it in a
Yes.
unified fashion. And you need to build a culture around that.
Yeah.
How did you prepare to, how did you gain that skill set?
So
I mean, you're very naturally very charming. I'm sure that played into it, but
I think that how did we get there? So I knew about the complexities. So I'm a first principle guy. I'm trying to sort of figure out the problem set that's in front of me and then try and find solutions. Doesn't mean that I always get it right. In fact, quite frankly, I think most of the time, any progress in Bit.bio has been based on failure. But that's also a thing, we celebrate, that we've learned something rather than that something has failed. So I had certain ideas. And then, of course, these were modified through reality and failure. One other thing that was very helpful at the beginning to really sort of put me on to a new sort of mindset. There was an accelerator program in Cambridge that I joined. So at least I had some understanding of the language.
Yeah.
And then the other thing that happened was very quickly the idea, the vision of Bit.bio is pretty bold. I mean, I think it can be incredibly transformational. And people have recognized this. So I was very lucky to be, backed by people that have done this before. So one of the first investors was Jonathan Milner. He built a company called AppCam. I had angels that had venture capital background, I really obviously I was drinking from the water hose, but I had an incredible support structure up until today. My board is, incredible. They're helping me to understand what I don't know. I just think you need to have a learning mindset, being open, and I love new stuff.
Yeah. And the community. It seems like building a community around you of people who've done it before, having that incubation experience.
That's right.
Accelerator experience.
Yeah.
Excellent. Typically, not typically, but more often than not, when we talk with cell therapy companies, their successes have been in rare and ultra rare disease. And Bit.bio strikes me as interesting because you're looking to tackle indications that are far more common, right?
Yeah.
What's this like? What was sort of the intent there, the strategy? Why is that?
I think when you think about the history of stem cell biology, it was always felt that this is the key that we need to unlock for regenerative medicine. So maybe 10 years back, 15 years back, cell therapy was always in the context of regenerative medicine. Then, of course, the world changed with the CAR-T field, which became an incredibly effective weapon against cancer. And I think we're only at the start of that. Now, Bit.bio has, embraced this regenerative medicine paradigm, and the way that we look at our internal pipeline is really trying to see what has worked. We need to de-risk clinical risk. If you think about your own pipeline, there's of course technical risk, execution risk, regulatory risk, etc. So if you take a risk-based approach on structuring your pipeline, what you want to see is patients that have benefited from a cell transplant, foremost. And these cells might have been donor-derived cells or autologous, so cells that come from the patients themselves. All of these approaches have massive constraints in terms of cost, in terms of scalability. And what Bit.bio the value proposition of Bit.bio is really that we can produce cells en masse and really democratize access to these therapies.
It's the next line of questioning I have for you was around that very thing. I mean, the cell therapy space has been shown so much promise.
Yeah.
But at the commercial stage, it's been a rough go, right?
Oh.
I mean, so unpack that a little bit for me. How the Bit.bio approach to addressing these indications shifts that cost, that incredible cost paradigm.
Yeah, at the moment, if you think of CAR Ts, it's really taking blood from late stage patients with cancers. You scratch these cells together, you then modify them genetically, you put them back, and you can already see how difficult this is. So the only way away from this is, at this moment in time, is an allogeneic paradigm, i.e. a centralized manufacturing capability. Now, the cool thing is that, and of course that raises questions about, how do we match the cells, how do we make sure that they integrate, etc. The cool thing is lots of progress has been made on this. So you can either, in the CNS, we just saw some beautiful data from Blue Rock Therapeutics. These cells persist after a year of treatment with immunosuppressants. So you've got a paradigm for the CNS. You have seen encapsulation, for example, in the context of Vertex pancreatic islet cells, curative treatment for diabetes. You've seen very enticing data from Sana, for example, in terms of the hyperimmune approach, where you take away surface epitopes and that cells can no longer be recognized. So this, together with a centralized manufacturing basis that can really create scale and consistency of highly functional cells, I think is going to be transformative. We think that we can lower the cost of goods of cell therapies by 2 orders of magnitude. We've got line of sight. And the reason why I can say that is because Meetable, they're moving to 500-liter tanks. They need to reach a cost of, a kilogram biomass cells, that's trillions of cells, for $10. Otherwise, they won't be commercially viable.
Well,
I can see them.
Sure. It was one of my questions about Meetable. Like, following on the heels of the cell therapy space, is the pork chop going to cost $3,000?
That's not going to work.
Right.
Exactly.
Right.
The crazy thing is they have a clear path to get there. And for a cell therapy company, obviously, you can add 2 zeros and you're still, you're in the realm of biologics. I feel very confident. And that's part of the disruptive power of this approach, that we can do that. We will do that.
Yeah. Where are you on the clinical sort of continuum?
Yeah, so we're preclinical. The earliest we can go into clinic is about 18 months from now. We have announced a pipeline. So the first product will be in the liver space. Again, as I said, based on something that worked really well in an early stage study. We have other cell types, pancreatic islets. We have various different immune cell types, CNS cell types. And of course, the company is at the stage where we want to augment, dilutive funding sources with partnerships. We're a classical new modality platform, very complex, very broad. And of course, if you think about the mRNA companies, what they've done is entered a lot of partnerships to get to the next stage where they become self-sufficient companies. So we've already announced one deal with Bayer, Blue Rock Therapeutics. We're going to develop Tregs together. And now JPM, of course, is a good conference to add to that partnership. pipeline.
Very good. You earlier you were talking about the breadth of talent and skill and the need to bring them all together. You talked about, like, beyond the lab manufacturing. What is sort of the manufacturing strategy of Bit.bio? Are you doing that internally?
So Bit.bio has launched research grade cells, which are now used by the top 10 pharma companies and considered as best in class for drug discovery and understanding human biology. So we've built obviously R&D grade manufacturing capabilities, and there are now significant portions now automized. So that's also been, part of the mission always is democratize access to human cells, close the translation gap, make sure that conventional drugs are being developed with more efficiency. And then, of course, on the clinical side, we have GLP at the moment, and we work with external GMP partners as we grow, with obviously in-house all of these processes.
So the process is in-house, but the intention would be moving forward to most likely outsource manufacturing.
At this point in time, we have to, but I think one of the key capabilities of Bit.bio is that manufacturing know-how. So I think that will be part of the business model, is part of the business model. So we just have to grow to clinical stage before we can, justify the investment at a larger scale.
When I look at the cost paradigm aside, when I look at recent cell therapy approvals, there have been plenty. You could write many case studies on commercial transition failure.
Yes.
Right. Like, I'm wondering, and I know it's early for you, you're preclinical, but I'm wondering, as the leader of Bit.bio, when do you start thinking about that?
At the beginning. Right at the beginning. Now, one of the things that has been really transformative for the company is when Catherine Corso joined us. She led cell therapy development at Takeda, very large portfolio, and she's one of the rare human beings that was able to create multiple products from scratch to commercialization. And she very clearly stated that we have to think about scale and the ability to industrialize our manufacturing from the beginning. So this is all baked in from the onset. I talked about it: single-step protocols, fully defined media conditions, 2D, 3D transition, and that's working extremely well. Plus all the processes, assays that need to come with it. So this is part of, I would say, the DNA of Bit.bio.
If you had to if you had to put your finger on a potential rate limiting factor or risk that could slow you down.
I mean, capital is one thing. Everyone has those constraints. I mean, obviously, now we are blessed in some sense that we have a revenue generating business, which is going to offset platform costs in the next 2 years.
What's the revenue generating?
So these are, so if you go to a website, you can order, for example, neurons, disease models. They have, neurons with Alzheimer's mutations. You have CRISPR-ready cells, which are essentially cells that you can put in your culture. You throw some guides in, you've got your experiment. So it's really about, one of the big things about stem cell is that it's so difficult to control. So we want to make it easy. Anyone can use them. You don't have to have any knowledge. You just take out the cell, a vial of, our microglia, of our, cells that are myocytes, so muscle cells. You've got, disease, Duchenne, disease mutations, etc. And that's how we connect again back to, the rare disease world, where, I think the rare disease world is incredibly rich in terms of scientific insight that you can gain. I mean, it's And of course, there's huge unmet clinical needs.
Yeah. So getting back to the, I
Sure.
have a tendency to just go down rabbit holes and take you off there. But getting back to potential rate limiting factors,
yes.
you talk about capital.
So capital is one thing, of course. What else could happen? We can have, in the clinic, all sorts of things can happen, from not being able to recruit patients to halts that are possible. So we're quite aware of all the risks there. What else could happen? I think there are risks around. I don't think we have technical risks any longer. So one thing that we've learned over the last 4 or 5 years is how robust the technology is. In fact, I'm going to just throw out something. We've not had a single manufacturing failure in the last 2.5, 3 years in a cell therapy product. We've done other things that went wrong. We didn't pack the right number of cells in the vials for our customers. We mislabeled vials, but we've never had biological failure. So that's very different, I would say, from any of the cell manufacturing company.
Yeah. You mentioned, hinted at regulatory, right? The
Sure.
regulatory landscape. What similar to starting to think about commercial and making a hire to make sure that you're, beginning with that end in mind. At what point does regulatory expertise come into play?
It's of course very early. You have to think about your target product profile, the path that you want to navigate through the regulatory landscape. Of course, rarer conditions have an easier path, so that's going to be the entry, also for our first cell therapy effort. I've lost my naivety, I would say, and I've got a team that has, all of these things covered. I'm very proud of my team. I mean, just think about who sits on our board. Greg Winter, Sir Greg Winter, Nobel Prize for his antibody technology. I think multiple companies. I think 2 years ago, the 3 best-selling products were based on his technology. So he's seen a lot of nonsense and things that can go wrong. So I feel that the board as a whole is an incredible resource that helps us think through what could go wrong. And I've got a very experienced management team.
What's your what's the funding strategy been to date? We're gonna talk a little bit about what it looks like going forward. I want to get your sense for the sentiment of the market at this point. But since founding, what's been your
the trajectory we span out? I sort of had this idea, and it was a single person idea at the beginning, and we raised a small amount of pre-seed from people that know what they're doing in the Cambridge ecosystem. Then very quickly after that, we had an early stage investor. I think fantastic capital, deep tech / biotech called Blue Yard. I always refer to it as US money in Europe, super supportive. And then in another year later, we struck really lucky. I was able to get Rick Klausner, Bob Nelson, and Jim Tannenbaum, so Arch Ventures, Foresight Capital, and Yuri Milner, Milky Way, Rick Klausner interested, and they led our Series A, and that was a big splash. Really allowed us to, at that point in time, they contributed more than 40 million. That allowed us to, build the platform, understand how we could create something that can create new cell types and industrialize the creation of new cell types. And then we at the end of 21, we raised a larger financing round, 140 million, and that was again led by Blue Staples, a combination of insiders of strategics, Charles River, National Resilience, etc. So that's given us a good capital base that allowed us to build this. I think we were quite outstanding when you look at the UK landscape. And actually, we followed nearly a textbook approach. You think about platform, new modality platform, it takes a lot of capital to build it. So we're now there. I can say the platform is fully validated end to end. Of course, proof of concept in the clinic is still in front of us. At that point in time, we then have to think about non-dilutive funding opportunities. So we talked about our deal with Buruk and Bayer and our ambition to really increase that now. And we've got a real pipeline of interest, and that's very positive. And of course, we will need further funding in order to drive the Bit.bio to the next level. At the same time, I think from a company size, from a maturity sort of perspective, we're reaching a point where maybe in 18, 24 months, we'd be ready for an IPO. So this is not to be seen as an exit. I think this is a transition from one capital pool to another capital pool. We've done our homework. We've got all our systems in place. We could jump anytime if necessary. But I do also think that now is the time, and we're sort of actively bringing investors together to finance another round that then sets us up for that next stage.
That next stage being the clinic.
The clinic and potentially the transition into public markets.
Gotcha. Okay. So what specifically is your strategy when you come into an event like this?
We've obviously lined up a lot of investor meetings. We already have started to engage them since November. There's been education earlier on last year. I think we're in a very good spot at the moment. And, we've got our ambition to, crystallize this in the very near future. So,
I want to just jump back to Meetable real quick because I'm curious about from a leadership perspective. You're obviously leading Bit.bio and Meetable. You found it.
I found it. I'm nearly at arm's length. There was a very strong tech collaboration at the beginning. Naively, again, I said, I can translate this into non-human species. It took a bit more than the idea, but I found, the teams really sort of were able to do it, and then Meetable took it and ran with it, and now they've got an excellent technical team there, and I've just visited their facilities and tried some of the products, and it's extraordinary, actually. Yeah.
How close to commercial is that?
So in terms of they need to get to that next scale point, which is now no longer a biological question. It's really about having access to the infrastructure and solving some of the things, the distribution of nutrients in the bioreactor. That's looking extremely, extremely good. And then they are launching, let's say, early access in Singapore. They're eyeing up the US now as well. I think the regulatory framework in Singapore, of course, is incredibly conducive. In the US, it's also very positive now. And then once this is demonstrated, I think that's when the capital will be available to really create the commercial scale facility that will drive down costs to the point that it's competitive.
Yeah, that's super interesting. What was the third company you said that you founded?
Bit.bio. It's really a take on a unique property of pluripotent stem cells. They reside in, you could say, a state of perfect homeostasis. They can buffer all the entropy, the things that go wrong or at every moment in time, and repair it themselves. But the problem is, once they exit this pluripotent state, age starts to come in. The repair mechanisms, and I think potentially by design, are being turned off very slowly. And that's the reason why, we get gray hair, because we have all these sort of things that sort of change as we grow a little bit older. And so we found a way of figuring out how stem cells maintain this useful state. We have a complete, Bit.bio is trying to create a complete map of all genes that are involved in the regulation or the repair of what's called the aging hallmarks. So, and then again, Bit.bio and Bit.bio, both of them are nearly hypothesis-free, pure data plays where, large data processes, ML, play a role. Same for Bit.bio. All we want to understand every gene and its relationship to these aging hallmarks, and then use that knowledge to create rational choices and prioritizations for interventions that can deal with age-related conditions, or maybe even extend the health span as such. So you could see this is the flip side of the coin. If something went wrong, you need severely wrong, you need Bit.bio, making sure that things don't go wrong or preventing that time, pushing it out, that's Bit.bio. And the magic in the middle is the purpose.
What is your engagement level with Bit.bio?
So we discovered the approach in the academic lab, and now I've handed it over again to a team. So I'm on the board. I'm curious of what they're doing, but I'm not involved in the day-to-day management.
Okay, yeah, I was curious
That's
about
not
that.
possible.
I mean, the anti-aging space. I mean, it's a I'd say nascent, but it's
coming.
the corner. Right?
I think the biology is solid, and it's very interesting.
What has been the most on this trip to San Francisco? What's sort of opened your eyes the most in terms of what you're hearing and seeing as you traverse the streets and have your meetings at JP?
Look, I've been very focused on Bit.bio meetings, so partnership meetings, investor meetings, and what my take is that Bit.bio has now reached a visibility that we didn't have a year ago. So you go somewhere and actually people say, "Oh, Bit.bio, I've heard of you," and of course we need to amplify this. That's an important thing. The validation that the partnership with BlueRock has provided has also dropped into people's minds. So if you think about it, there are really two big companies. We talked about them, Vertex, Bayer/BlueRock, that know something about this space. And I think the signal that one of them has asked us for help has really sort of changed the perspective. And so I think that's extremely positive. And on the investor side, look, there seems to be a better, the weather seems to be better. And I think that's true literally when you think about the torrents of last year.
Miserable.
No, and we can, there's an uptick in terms of, mergers and acquisitions. There was good news flow in terms of financing. There's a number of companies that are going IPO. I'm really sort of keen to figure out how metagenomics, for example, is going to fare. It's very exciting. I think we've turned the corner. That's what we sense. Is it going to be smooth sailing from here? We don't know, but let's take the momentum that exists, and our objective is really to crystallize the round now in Q1.
Yeah, excellent. Well, I wish you luck with that, and I appreciate the fact that you set some time aside during this very busy week to spend with me and the Business of Biotech listeners. So Dr. Mark Kotter. It's an honor, a pleasure, and we'll be following along. All three of these companies are very fascinating and in really good positions. So we'll be following along, and hopefully we'll have an opportunity to meet with you again down the road.
Thank you so much.
Clinical stage. I'd love to get some clinical updates when you're.
I'm looking forward to that as well. Although I think, on the other side, the use of cells to create better drugs, it's also very, very exciting. Well, thank you for having me.
Thank you for joining me. Appreciate it. And safe travels.
Thank you.
I'm Matt Pillar, and you just listened to the Business of Biotech, the weekly podcast dedicated to the builders of biotech. We drop a new episode with a new exec every Monday morning, and I'd like you to join our community of subscribers at bioprocessonline.com, Apple Podcasts, Spotify, Google Play, or anywhere you get your podcasts. You can also subscribe to our never spammy, always insightful monthly newsletter at bioprocessonline.com/BOB. If you have feedback or topic and guest suggestions, hit me up on LinkedIn and let's chat. And as always, thanks for listening.